Josie. Recording in progress. Okay. All right, to all the participants, it is my pleasure to introduce Dr. Josie Ursini-Siegel, who is our invited speaker today at the QCRI seminar series. I'm sure several of you know her already and her accomplishments as well in the field of breast cancer research, and I would like to very briefly highlight some of her contributions here. So Dr. Ursini-Siegel is currently the Director of Molecular Oncology Group and Associate Professor at the Departments of Biochemistry and Oncology, Lady Davis Institute for Medical Research at McGill University. She completed her bachelor's, master's, and PhD degrees at McMaster Universities and did her postdoctoral fellowship at Wales Medical College of Cornell University and McGill University. She's been awarded over $7 million in competitive funding from multiple organizations and has published 32 papers, including 12 as corresponding author, 5 review articles, and 15 co-authorships in very high impact journals such as Nature Communications, JCI Insight, Cancer Cell, Cancer Research, Oncology, and others. She runs a multidisciplinary and collaborative research program that is focused on understanding the molecular mechanisms driving the evolution of treatment refractory and metastatic breast cancers, and to identify novel treatment strategies to improve outcomes for these women. So using multidisciplinary approaches, including mouse genetics, molecular and cellular biology, and omics-based technologies, her team is specifically investigating specific questions on the tumor immune microenvironment, deregulated activation of mitogenic signaling pathways, oxidative stress, and metabolic reprogramming. And most recently, her group has published a study in Nature Communications demonstrating STAT1-induced inflammation sensitized breast cancer to the cytotoxic effect of Biguanides by inducing energetic and oxidative stress. I think this will be a focus of today's talk as well. So Dr. Siegel also serves on the editorial board of Methods and Molecular Biology and on several grant panel review committees, which is where I met with her and have heard her great contributions, including CIHR, CCSRI, CRS, and others. She's also the recipient of the New Investigator Salary Support Award from CIHR. And I definitely am not going to pronounce this correctly, Josie, so apologize. I apologize for that. And it's the Senior ShCA Award from, I'm so sorry if I mispronounced it. I tried and I couldn't find it. From McGill University. As I mentioned, her research program is funded by several federal and other cancer agencies. Dr. Siegel, welcome to Queens, welcome to Kingston, and I really wish you were here in person. We are very excited to hear about your research.
That's very kind, Madhuri. Thank you very much. That very kind intro and the introduction. It's really wonderful to see some old faces and new faces, and thanks for the opportunity. Well, I'll go ahead and not sure I can screen share. Let me see. You might have to make me a. You guys can see that?
Yep.
Okay, awesome. Okay, so please feel free to interrupt at any moment. I'm sorry. There we go. Okay, so there. Okay. Oh. Okay, so again, thanks for the introduction, and it's a real pleasure to be here. And today I want to talk to you a little bit about research that we've partaken over the last few years. Most of it was published recently. I'm going to give a little bit of sort of historical perspective on some of our earlier work that led to these observations, and then some unpublished data at the end. Again, feel free to interrupt me at any time. So as Madhuri mentioned, my interest is really in trying to identify novel, more rational therapeutic strategies to treat women who are affected with breast cancer. And my reasons are several-fold. So first of all, just to briefly summarize, these are the treatment options that are currently available for women with breast cancer. So we have surgery, chemotherapy, radiation therapy, and as well as targeted therapy, including endocrine therapies or HER2 targeted therapies. And the choice of therapy really is dictated by, I would say, two major criteria. One is subtype, which is basically defined by the expression or lack of expression of three molecules, which is estrogen receptor, progesterone receptor, and or HER2. And the second is stage and grade at which the individual gets diagnosed. And that really is the sort of go, no go points in terms of the therapeutic options that a patient has. And another point that's important to make is that although these targeted therapies really have revolutionized and really improved treatment outcomes for women who are within the correct subtype and get diagnosed at an early stage, for women who get diagnosed at a later stage, relapse is either more likely or inevitable if it's metastatic disease. So there's still a lot of room for improvement, and that's where my research program focuses on. So the standard approach for treatment really focuses on the genetic heterogeneity of tumors. And this is something that I'm sure that you're all familiar with the research that is ongoing in your groups, but the idea that there's individual clones that each have their own genetic makeups. And following relapse, it's understanding the genetic differences that predominate in the majority of the clones. If you can identify a targetable pathway, then that gets treated, and then you eventually get secondary resistance and so on and so forth, leading to the concept of heterogeneity. But the limitations to this in terms of treatment opportunities are the fact that there's significant plasticity in aggressive breast cancers which drive resistance. And again, I'm sure this is something that's not new to the audience, so I'll summarize it briefly. But plasticity not only, on several fronts, right? One is genetic, which we talked about, genetic and epigenetic differences. The second is that even within an individual patient, not only are there genetic differences within the makeup of an individual tumor, even the tumor and the metastatic lesions are entirely different. So it's very hard to treat a whole body in terms of cancers that are spread in women. And the third are profound changes in the tumor microenvironment that can really alter the responsiveness or lack thereof to particular therapies. And this includes changes in nutrients, growth factors, oxygen, immune infiltrates, and how cells integrate these cues outside of any genetic changes. So keeping all this in mind, it's clear why the current therapeutic approaches there's limits in terms of what they can offer. So more increasingly, as I've progressed in my research, I'm really starting to think about this a little differently. And the idea that instead of taking these targeted approaches, the better approach is that we've identified targetable essential vulnerabilities as a strategy to bypass this tumor heterogeneity, which we recognize is a limitation. And I just summarize here some of the what I would consider essential vulnerabilities that I think are worth exploiting. And there's many clinical trials exploiting these different targetable pathways. I'm boxing, so in green are the three that I'm going to touch upon today: the concept of metabolic flexibility, oxidative stress, and chronic inflammation. And at the end, hopefully, I'll bring this all together for you. So I'm going to start with metabolic flexibility. And what is metabolic flexibility? Well, it means the ability of a cancer, and these are typically more aggressive breast cancers, to utilize numerous different kinds of nutrients to meet their either energetic or biosynthetic demands or raw scavenging potential. And by doing so, by engaging different metabolites, they can either, I'm not sure if you can see my pointer, but generate metabolism through glycolysis, which is outside of the mitochondria, or through oxidative phosphorylation of many, many metabolites, including glucose, amino acids, lipids, so on and so forth. So tumors that have this level of metabolic flexibility tend to be the most resistant to the therapies that I introduced at the beginning. And there's been a lot of research over the last few years to try to identify, can we actually exploit combination therapies that target both glycolysis and oxidative phosphorylation? And is this a new therapeutic strategy to overcome resistance? I'm just going to introduce this class of drugs that's relevant to my talk, Biguanides. Biguanides, so these are anti-diabetic drugs. Metformin is one that we hear about all the time. It's used in the clinic to treat type 2 diabetes. And the sister molecule called Phenformin, which is the one that my lab predominantly studies. So mechanistically, on a whole body level, what these anti-diabetic drugs do is they increase glucose uptake. So it makes sense why they're anti-diabetic. But on a cellular level, they actually do so by blocking complex 1 of the electron transport chain right here. So this is the entry point of the reducing equivalents from the TCA cycle. By this, the transfer, this is also called the NADH dehydrogenase, and the flow of electrons from complex 1, 2, 3, and so on leads to ATP production. So Biguanides, by blocking this, significantly reduce the ability of the mitochondria to generate ATP, and therefore inactivate AMP kinase, which induces energetic stress in the cell. Okay. Most cells will compensate by increasing glycolysis, and this becomes important later. So why repurpose Biguanides in oncology? Why is this a good idea? This really has sparked a lot of interest, I'd say, over the last decade or so. The reason being, number one, these drugs are well tolerated and being used in people. So we know that hurdle has been met. But more importantly, cancer cells should have a selective vulnerability to Biguanides relative to normal cells. And the reason being is a cancer cell is more highly proliferative, has a much higher ATP demand, therefore is much more dependent on its mitochondria and OXPHOS. So that should create a therapeutic window of opportunity by which cancer cells are selectively vulnerable to Biguanides, whereas normal tissue is not. So that's the idea of introducing these into the clinic. And I'm just going to discuss very, very briefly, because this is published, some preliminary, some earlier studies that we did that I think point, at least in the preclinical models, that there could be something to this, and that the metabolic heterogeneity and flexibility of the tumor likely dictates Biguanide sensitivity. So for those of you who are familiar with my research, you know that I study receptor tyrosine kinase signaling, and particularly my lab focuses on an adapter protein that is important for transducing mitogenic signals from RTKs. It's called ShCA. For the purpose of this talk, all you need to know is that ShCA has 3 tyrosine phosphorylation sites that become phosphorylated by an activated receptor tyrosine kinase. And this phosphorylation event is required to transduce mitogenic signals. So what we showed in this paper in 2018 was that an intact RTK ShCA pathway was really important for increasing the glycolytic rate and capacity in breast cancers. And in doing so, breast cancers were able to increase their metabolic flexibility because they were able to engage glycolysis to generate ATP outside of the mitochondria, but also relying on their mitochondria for ATP production from glucose as well as glutamine and other metabolites, increasing their flexibility. So if genetically we reduce ShCA signaling, what we show is that there was this very significant block in the ability of the cells to increase glycolysis, and they became entirely reliant on their mitochondria to support their growth and survival. So we hypothesized that this would create a synthetic vulnerability for biguanides, which block mitochondrial respiration. This was tested in vivo, just on the right-hand side here, where we treated tumors at a defined volume with phenformin and show that tumors that have an intact ShCA pathway are resistant to phenformin. But if you block ShCA signaling and you decrease this metabolic flexibility, they become selectively vulnerable to biguanides. And this was work done by a graduate student in my lab, Young Im. Young further went to test, okay, so we know that we can make a tumor grow slower. Can we actually also impact the neoplastic transition with biguanides? And is there a genetic component to this as well? So for this, we used a genetic approach, and we relied on my lab works a lot with the middle T mouse model. It's a model that mimics receptor tyrosine kinase signaling, highly aggressive and metastatic. And we also were fortunate to have obtained a series of ShCA deficient tumor and mouse models that contain various knock-in alleles of different mutant ShCA molecules. This was from the Posson laboratory. So this is actually middle T with 2 wild type ShCA alleles, or middle T in which one of the alleles contains a ShCA that's non-phosphorylatable. Those 3 tyrosines have been mutated. And what we actually showed here is, so you can see that these different bigenic models palpate at different times. So wild type middle T, you get a first tumor at about 60 days post-birth. And if you mutate one allele to the non-phosphorylatable, it shifts to about 90 days. But irrespective, if we actually wait until the first tumor palpation, which is what I'm showing here, and then we just put the mice on phenformin in the drinking water, what we were able to, what we were able to show is that only in mice where there was a reduced capacity to signal through ShCA, do you have a significant decrease in the number of glands that are transformed, and also a very significant reduction in the volume of the tumors at the endpoint. Highlighting the fact that potentially differences in the tyrosine kinome could influence the sensitivity of pre-neoplastic cells to biguanides, suggesting that this is that this is a nuance that we have to pay attention to when thinking about using biguanides in oncology. This was further supported and beautifully demonstrated by research from the Topasiewicz lab, Cell Metabolism paper in 2018, where he actually showed that pharmacologically, if you combine tyrosine kinase inhibitors and complex 1 inhibitors, so biguanide, you can profoundly increase the cytotoxic effects in numerous cancer models, including but not limited to breast cancer, but that there was a significant amount of plasticity that allows cancer cells to eventually overcome this. So in and of itself, there's going to probably limitations to this approach. And again, despite all of the promise of these preclinical studies, and there was a lot of excitement that was generated, when you actually go look at what was happening in the clinic, enthusiasm became dampened. So a lot of epidemiological studies that looked at women who meta-analyses who were diabetic and receiving metformin showed very conflicting literature, where some suggested a better survival rate and others showed absolutely no association in terms of incidence at all. And more recently, the Stambulik and Goodwin team ran the first phase 2 randomized control clinical trial where metformin was used in non-diabetic women who had metastatic breast cancer. And they showed absolutely no survival benefit, which really dampened the enthusiasm for real world practice, the practicality of metformin. So I think that there's two major limitations thus far that we have to overcome if we want to revisit biguanides in the clinic. One is the concept of metabolic plasticity, which I introduced. And the second is the therapeutic dose. And the therapeutic dose that we use in preclinical studies is 5 to 10 times higher than what diabetics receive. So we have to find rational combination strategies that allow us to overcome this plasticity, lower the therapeutic dose if we want to achieve real clinical impact with this class of drugs in oncology. And that's hopefully the research that I've done is hopefully one step forward along this. So I mentioned to you that the tyrosines in ShCA are very important for mediating resistance to biguanides, i.e. if they're phosphorylated in breast cancers by tyrosine kinases, tumors are resistant to biguanides. So what we've done is actually generated cells in which individual tyrosines were mutated to try to map the precise signaling pathway. And here you can see if you mutate the twin tyrosines right here, which is 239, 240, there's really no effect on biguanide sensitivity. But if you lose the capacity to signal through tyrosine 313, these cells now become exquisitely sensitive to biguanides. So we've mapped the pathway. And fortuitously, at the time, another student in my lab, Angela Ahn, had completed a paper demonstrating that tyrosine 313 is important to suppress STAT1-induced inflammation in cancer cells. And this is shown here that if you mutate both tyrosine sites, tyrosine 313 on both alleles, you get a dramatic increase in STAT1 level and phosphorylation in independent breast cancer models. So there are real consequences of these changes in terms of immune response to immunotherapy, which I don't have the time to go into here. But we asked the question, is this modification of STAT3 and the ability to increase STAT1 coupled to the metabolic differences that we observe? This is briefly, before I get into this, I'm sure everybody here, knowing that Maduri works on STAT1, understands the signaling pathway, but just very briefly, tyrosine kinases can activate STAT1, which can homo or heterodimerize with other STAT family members, translocate to the nucleus where it functions as a transcription factor. And in breast cancer, STAT1 is thought to be tumor suppressive by its ability to modulate the immune microenvironment, induce anti-tumor immunity, but also stimulate either pro-apoptotic or anti-proliferative signals in the breast cancer cells. So we asked, is there an additional role for STAT1 in terms of controlling metabolic flexibility, given the observation that loss of tyrosine 313 in particular, which potentiates STAT1, induces biguanide sensitivity. So this was work that was done predominantly by a graduate student in my laboratory called Stephanie Totten. She's remarkable. And we decided we wanted to test directly if this was uncoupled from ShCA. So what we did was we took breast cancer cells that have very low levels of STAT1 activation and expression at baseline and just treated them with interferon gamma, which is the cytokine which activates the STAT1 pathway. So that's shown here. And what you can see here is an independent breast cancer models that if you combine interferon gamma and phenformin, you have a very profound sensitivity of breast cancer cells relative to either treatment alone. So this suggested that if you increase STAT1 signaling, you could induce phenformin sensitivity. And then we actually asked, well, is STAT1 important? So in these models, we generated STAT1 knockout versions. And what you can see here is that if you have wild type STAT1, you have phenformin sensitivity in combination with interferon gamma. But if you delete STAT1 by CRISPR in 2 independent models, we lose this. Okay, so this data together shows that interferon-driven STAT1 activation contributes to the increased sensitivity of breast cancer cells to biguanides. This is showing you that it's not a mouse-specific effect. This is 8 different breast cancer cell lines that span various molecular subtypes. And you can see here across all of them to various degrees, but in each instance, combination of interferon and fenformin is always more cytotoxic than either treatment alone. Okay, so now we wanted to get into understand the mechanism. So I told you that STAT1, a major role for STAT1 is in potentiating anti-tumor immune responses. And I showed you that if we reduce signaling pathways from RTK such that increased steady state STAT1 levels are increased in the tumors, that we can see increased sensitivity to biguanides. So we wanted to ask, if we manipulate tumor immune microenvironment, do we see similar effects? And I'm not going to go into it in too exhaustive detail because it's negative data and it was published, but basically just to show that we either took a genetic approach where we have CD8 knockout mice. They were syngeneic. We used an oncolytic virus approach in combination with fenformin, or we use an immune checkpoint blockade, so an anti-PD-1 monoclonal antibody in combination with fenformin. And in none of these instances did we see synergy with the combination. If anything, the effects are additive, suggesting that anti-tumor immunity does not, we don't think, underlies the sensitivity that we observed in vivo to fenformin. So then we asked the other way around. Well, is this about chronic inflammation? Because STAT1 is going to induce an inflammatory response. And to model chronic inflammation, what we decided to do is to treat tumor-bearing mice with a toll-like 3 receptor agonist, it's an adjuvant called poly-IC. It's a double-stranded RNA mimetic, and this potently activates inflammatory responses, including STAT1. And what you can see here in 3 independent models, these are 2 immunocompetent models. This is middle T in FEB, the 4T1 model in valve C. And interestingly, as well as the 231 model, which is a model of human breast cancer, which must be xenografted into immunodeficient mice, we saw that a combination of poly-IC and fenformin synergized very beautifully in all 3 systems to induce a cytotoxic effect, demonstrating the applicability of this combination therapy in multiple tumor types, and also highlighting the fact that it likely relies on innate immunity, because in this model, we have no more adaptive immunity. I'm not showing it here, but we also showed, at least in the middle T model, that if middle T cancer cells don't express STAT1, you lose this sensitivity. Okay, so together, these data suggest that it's inflammation in the tumor microenvironment that is sensitizing the cancer cells to fenformin. So now we wanted to understand the mechanism. And I told you that biguanides, their mechanism of action is to induce energetic stress by blocking complex 1. So is it possible that interferon gamma further potentiates this energetic stress and just basically pushes the tumors over the edge? So we tested this in several ways. And this was done with Julie St. Pierre, who's a close collaborator at the University of Ottawa. And this is negative data that I think shows quite convincingly that relative to fenformin, which profoundly blocks mitochondrial respiration and increases glycolysis, interferon gamma treatment on its own very modestly impacted these parameters. And the combination treatment really had no difference. You can see this here in terms of total ATP production, either from glycolysis or oxidative phosphorylation, that fenformin really profoundly reduced ATP mitochondrial production, led to a corresponding increase in ATP from glycolysis, which we would predict. And interferon gamma as a monotherapy really didn't change bioenergetics of cancer cells. nor did it change the production of glycolytic intermediates, suggesting that this increased sensitivity that we're observing likely doesn't result from changes in the bioenergetics of cancer cells. So we thought about this a different way, and maybe this has to do with ROS. And this is something that field recognized a long time ago, but we didn't pay attention to. So when you block complex 1 of the electron transport chain, by preventing the transfer of electrons from complex 1 to complex 2, what you're effectively doing is building up electrons in complex 1. So that prematurely these electrons can be transferred to oxygen in the mitochondria to increase the production of superoxide anions and the production of reactive oxygen species. And this is probed for mitochondrial ROS, showing that in 2 independent models in our system, phenformin profoundly increases ROS production. So the question is this increased ROS production at the heart of the sensitivity? So just to briefly summarize, what are reactive oxygen species? As I mentioned, they're free radicals on oxygen-containing molecules with unpaired electrons. And it's with ROS, it's not about all or none, it's really quantitative differences. So cancer cells, if they can have moderately increases levels of ROS that they can cope with, this increased ROS in the chronic state can actually increase changes in the cell that are tumorigenic. That's another part of my lab, but not something that I'm going to focus on here. But if you can find a way to push reactive oxygen species higher over this sort of moderate limit, what you do is you get severe damage of macromolecules in the cell, so that's protein, lipids, and nucleotides, and you can tip the balance to favor either cell cycle arrest or apoptosis. So if you can make ROS levels high enough, it can be anti-tumorigenic. This is just to show you that in our tumors, where we treated tumors with a combination of poly-IC and phenformin, we observe increased death. So this is caspase 3, specifically in combination treated tumors. And this was associated with increased oxidative damage of DNA. And there's a marker called AOXO-DG that specifies this. So it's the first indication that maybe ROS was involved. But when we measured ROS levels, I was surprised to see this, but we actually don't see changes in terms of total mitochondrial ROS production, either with interferon gamma alone or with the combination therapy in tumor models where the combination of interferon and phenformin clearly is anti-tumorigenic. So, okay, so we had that observation. So we thought, okay, let's just functionally test this. Is mitochondrial ROS important? And the cell controls ROS levels by, it's really a seesaw, as you can see over here. So increased metabolic rate increases ROS production. So as I showed, mitochondrial metabolism or the block in terms of complex 1. And the cell combats this by producing, generating a whole slew of different ROS scavenger molecules, which can effectively serve as electron donors to neutralize these free radicals. And it's the balance between these two that dictates whether or not you're going to have oxidative damage. So there are pharmacological drugs that can be used to neutralize ROS. We chose one called Mitotempo because it's specifically targeted to the mitochondria and serves as an electron donor, so ROS scavenger. So we asked if we treat breast cancer cells in vitro or breast tumors in vivo with Mitotempo, does that change the sensitivity of these tumors to a combination of phenformin and interferon gamma? And this just shows in 2 independent models in vitro that indeed this is the case. As you can see here, we can completely reverse the sensitivity. And in vivo, in terms of the tumor growth kinetics, we observed the same phenotype. So these data argued that mitochondrial ROS is underlying the sensitivity of these tumors to interferon gamma and phenformin. So knowing that total ROS levels didn't change with the combination treatment, what it led us to believe is that maybe what this combination, what interferon gamma was doing was decreasing the ROS scavenging potential of the breast cancer cells so that you can now increase oxidative damage in the cancer cell. So before we actually, I go in just to show the genetics in terms of the gene expression changes that we observed, we wanted to test this directly. So if we manipulate with the ROS scavenging potential of a cancer cell, is that sufficient to induce sensitivity to phenformin? And we chose to look at glutathione. Glutathione is the most abundant ROS scavenger in all of our cancer cells. It's a molecule, it's 3 amino acids that contains a cysteine, and the cysteine can act as an electron donor. And there's a drug called BSO, which blocks the first step in glutathione synthesis and is also being tested in clinical trials, cancer clinical trials. So we wanted to ask, okay, if we block the ability of the cells to make glutathione, can we sensitize them to phenformin? And this is just showing here a dose response curve showing very profound sensitivity between BSO, which on its own had no effect, and increasing concentrations of phenformin. And this is showing that if we reverse mitochondrial ROS production, ROS production with mitotempo, we reverse this sensitivity to the combination treatment of phenformin and BSO. So simply by blocking glutathione synthesis, we can potentiate to phenformin sensitivity. So we looked at the glutathione pathway in response to interferon, and it was not really changed at the level of expression or the GSH-GSSG ratio. So this couldn't be the mechanism. So we decided to perform before I jump to that. This is a computational study that we did. So all of the data that I'm showing you is in the context of breast cancer cell lines in terms of models. So we wanted to ask, are these observations also being observed in human breast cancer? So this is the TCGA database. And what we did here is it's a computational approach that we do with Claudia Kleinman. So we generated gene signatures of either STAT1 target genes, and it's a 90 gene signature, and that's on the y-axis, or an oxidative stress gene signature. It's a 97 gene signature, and that's on the x. So the oxidative stress gene signature refers to genes that are transcriptionally induced when a cell receives oxidative stress. Okay? And we wanted to ask, and this is called a single cell gene set enrichment analysis. So what it determines is how much are these signatures over or underrepresented in individual tumors. And what we observed is in all breast cancers, irrespective of subtype, a linear relationship between whether or not the cell has its STAT1 transcriptional response and is likely dealing with oxidative stress, which is correlative, but I think is promising. So now into mechanism, we wanted to understand at the mechanistic level how interferon gamma signaling through STAT1 could be potentiating oxidative stress. So we ran an RNA-seq experiment in STAT1 wild type or knockout breast cancer cells. And we identified a family of genes that are important for ROS scavenging, both pro-oxidants as well as antioxidants. And one caught our attention called NQO1, which was significantly diminished in STAT1 wild type cells relative to STAT1 knockout cells. Okay? This is just validating by QRT that this is correct. So if you treat cancer cells with interferon gamma, you reduce steady state NQO1 levels, and this is STAT1 dependent because if you CRISPR STAT1, you lose this. And And this is just showing you that if we treat a variety of human breast cancer models with interferon gamma, in some but not all, we actually observed it in 3 of the 8. You have decreased interferon-induced expression of NQO1 at the protein level. So what is NQO1? NQO1 is a 2-electron donor. It's a quinone reductase. It's in the cytoplasm of cancer cells, very frequently overexpressed in cancer cells, and can serve as a ROS scavenger. So genetically, first we wanted to ask, can NQO1 substitute, knockdown of NQO1 substitute for interferon gamma? Or does NQO1 overexpression confer resistance to the interferon-fen combination therapy? This is showing the overexpression, showing that yes, indeed, you lose the sensitivity observed if the cells have ectopic NQO1 overexpressed, and that if you knock down NQO1 in breast cancer cells without interferon gamma, you phenocopy the effects of what we observed with interferon gamma co-treatment. This is in 3 independent models. We show this, and here we're actually showing again using mitotempo that this increased sensitivity that we observe if you knock down NQO1, combine it with fenformin, is mitochondrial ROS dependent because we can reverse this with mitotempo. Okay, so how can we exploit this pharmacologically? There's a drug that's in several clinical trials for solid tumors called B-Lapachone. So B-Lapachone is actually a substrate for NQO1, and in NQO1 positive tumors, actually uses NQO1 to generate free radicals itself. So it not only does B-Lapachone become a free radical with NQO1, but NQO1 in this instance is basically, I'd like to say, being kept busy. So it can't neutralize endogenous sources of ROS. So we asked the question, does beta-Lapachone synergize with fenformin? And this is just showing you in vitro that indeed, yes, it does. In vivo, we also see increased synergy of beta-Lapachone in fenformin, and that this is associated with increased oxidative damage of DNA in the combination treated tumors, which I'm showing here. So beta-Lapachone can substitute for poly-IC in terms of increasing fenformin sensitivity. I'm going to skip this for the sake of time. And this is just showing you in several PDX models of HER2 and triple negative breast cancer that a combination of BSO and fenformin or beta-Lapachone and fenformin on the bottom can synergize, at least in vitro, to induce cytotoxicity, hopefully showing the generalizability of these observations. So for this part of the talk, this is just sort of a summary of the model that we have. So breast cancer cells, although they are more reliant on their mitochondria for ATP synthesis, and fenformin is able to induce energetic stress in breast cancer cells. Under these instances, this is primarily cytostatic because the cells, number one, can compensate by increasing the rates of glycolysis, so they don't have severe energetic stress, and likely also increased expression of ROS scavengers to combat the extra ROS that's produced. So the idea is, can we exploit this increased ROS therapeutically? And what we show is that either through interferon-driven STAT1 inflammation or directly using drugs that target the mitochondrial ROS scavenging machinery, that if you can simultaneously induce energetic and oxidative stress in these cancer cells, you now have a cytolytic response that confers much stronger anti-tumor properties. And the final part of the talk, just this, not too many slides. This is unpublished data. It's preliminary data, but I wanted to show it so that you guys have a sense of where we're going. And I think that there's possibilities for collaboration with several people on this call. So what I showed you now was the context of what's actually happening in the tumor cell if we stimulate inflammation and why you have increased sensitivity. But what we really don't understand is how is over-3 coupled inflammation actually potentiating all of this, right? So this is just a reminder to you that toll receptor 3, poly-IC is an agonist for toll receptor 3, activates numerous inflammatory pathways in immune and non-immune cells, and can exert anti-tumorigenic properties through multiple mechanisms. One is inducing inflammatory signals by producing inflammatory cytokines. The second is by eliciting anti-tumor immune responses, both directly and indirectly. So directly by activating innate immune cells that have cytotoxic properties, and indirectly by activating cytotoxic T lymphocytes and NK cells. So the work that I'm going to show to the end is by Joey Heath, who's a graduate student in my lab. So the first question we asked was, if you take control-treated tumors, so placebo, or the poly-IC Fen-treated tumors where we activated Pol-3 with the biguanide, and this is the 2 independent syngeneic models that we had. By flow cytometry, who are the immune populations that at least numerically are changing? And the ones that caught our attention were either CD4 positive T cells or Ly6G positive neutrophils. And here you can see one of the 2 tumors models, the 4T1, you have increased recruitment of CD4 positive T cells. And here, both of them, you see recruitment of Ly6G positive neutrophils. But if we included a dead cell stain and track the cancer cells using a CD45 negativity, what you observe here is no real association in terms of viability and the density of CD4 infiltration. But on the right-hand side, in both tumor models, there was a very strong negative correlation between infiltration of neutrophils and viability of breast cancer cells. So this really focused our attention on the neutrophils in terms of their increased recruitment and whether they were contributing to the increased sensitivity to complex 1 inhibitors. We showed using Ly6G neutralizing antibodies, and these specifically deplete neutrophils in vivo, that you can reverse the sensitivity of poly-IC Phenformin treatment. This is in the 4T1 model. If you pre-treat the mice with neutralizing antibody to neutrophils, saying that this immune cell type is important for the cytotoxicity. And this is kind of where we're going now in terms of looking at the model. There's 4 possibilities. One is that you have indirect effects in terms of the neutrophils, in terms of increased sensitivity. One is their ability to release pro-inflammatory cytokines. And the question is it these inflammatory cytokines that are then stimulating STAT1 in the cancer cells to induce oxidative damage? This one, I think is lowest on the list because we show that adaptive immunity is not important, but neutrophils can serve as antigen presenting cells. But interestingly, neutrophils are one of the immune cell types. Part of their mechanism of action is actually to increase the production of ROS. So on their cell surface, they contain NADPH oxidases that produce superoxide anion, which can then be released as part of the respiratory burst into the microenvironment. So the question is, are the neutrophils directly potentiating ROS by increasing the secretion of ROS into the microenvironment? I have no data on this yet. This is where we're going, but I just wanted to present it to you. And this is just preliminary data that we have that I'm kind of showing for Lynn because I know that she has great interest in an integrated stress response, but we started to look here. And why we focused on this is, so we know that STAT1 one of its target genes is PKR, and PKR is one of the kinases that can induce an integrated stress response by phosphorylating eIF2 alpha. And toll receptor 3 is actually a double-stranded RNA mimetic, and a poly-IC, sorry, is a double-stranded RNA mimetic, which will recognize toll receptor 3 and activate PKR. So we wanted to ask the question, was there anything to this? And this is just showing that the combination treatment, in fact, does induce phosphorylation of eIF2 alpha in vitro. And again, this is just in vitro, and it's just one model, preliminary data, but what we're actually showing is that if you can block an integrated stress response using ISRIB right here, you can reverse the sensitivity to interferon phenformin treatment. And if you treat the cells with an agonist that potentiates an interferon gamma stress response, you increase the sensitivity, which says there could be a component of an integrated stress response to the regulation of this sensitivity. And this I found extremely interesting. So we actually went in and asked, what about in the tumor microenvironment? So again, by flow cytometry, we just looked at phospho-eIF2 alpha levels in the various immune cells. And this is just showing you here that in the neutrophils, although we didn't actually observe changes in total levels of phospho-eIF2 alpha in the control versus combination drug-treated tumors, in the tumors where, if you look at the neutrophils, there's an inverse association between phospho-eIF2 alpha mean fluorescence intensity in the neutrophils and tumor viability, which was intriguing to us. So genetically, this is the last piece of data. We tested this. We collaborated with Antonis Koromilis, who has a germline mouse model where one of the two alleles cannot be phosphorylated by eIF2 alpha. So it can't initiate, it has a reduced capacity to induce an integrated stress response. And this is just showing you that these mice now become refractory to the combination treatment, whereas wild type mice are sensitive. So this is just showing you that we think that an integrated stress response, both in the breast cancer cells and as well in my tumor microenvironment, we're postulating it's the neutrophil, but we're not sure, contributes to the sensitivity of toll receptor 3 inflammation combined with complex 1 inhibitors. And this is something that we definitely want to explore. I have some ideas in terms of models, but we can talk about that later. So this is my very last slide, just to show you, tying it back to the beginning. I said to you that I think the next approaches that we should be targeting are about using targeting essential vulnerabilities. And what I'm hoping I convinced you of is that inflammation combined, so an inflammatory stress combined with an energetic stress, we use Biguanides, really touches upon 3 essential vulnerabilities: metabolic flexibility, oxidative stress, and chronic inflammation. And although it's a preclinical study, and maybe the exact pairwise combinations isn't the right one, that hopefully this approach can have some traction in the clinic if we move it forward. And my last slide, of course, thanks to people. This was a lot of work that really took a lot of effort. I tried to thank the people along the way. And for the sake of time, I'll leave it there. And funding, of course, and thank you for your time. And I'll stop sharing.
That was a fantastic talk, Josie. I'm sure everyone enjoyed. That was really fascinating work and very in-depth. And I think it really emphasizes how we should be incorporating multiple models and different approaches and really going to the root. Before I open it to others, I have a couple of questions because of the interferon pathways and STAT1 roles. And STAT1, I think, as you have also shown, it seems to be paradoxical type 1, type 2 interferon response. In breast, as per my knowledge, there is basal and luminal kind of classification. And in bladder, I know basal tumors are heavily infiltrated. Do you think that pre-existing immune infiltrate state will somehow modulate